helps help solve some of the questions we have. And so, and everyone is aware in this room, the diagnostic odyssey was probably a huge challenge. And now, you have a molecular cause of your child's condition. But from my point of view, even for new disease genes or recently identified disease genes, we still have a lot of variants of unknown significance in these same genes, and particularly for missense changes. These are changes in the genes where it just changes one amino acid to another type, and they're very hard to figure out, even though clinically we may have kind of solved the diagnosis. So, testing variants and using model organisms to do that, to actually show and prove that it's disrupting protein function, is a powerful tool. And so for many who I talk to, of course, they ask me if I need the fruit flies from their kitchen in my lab, but those are not the flies we want to play with. We've made genetically modified flies. But if you're not aware, for the last 100 years, the flies have basically been first for discovering fundamental genetics, signaling pathways for the scientists in the room, wint, notch, hedgehog, toll, etc., developing models of human disease, some key fundamental neurobiology, and importantly, can be used for drug screening very quickly. But only in the last 10 years or so, since we've now entered the era of precision or personalized medicine and then identified a lot of these genes and genetic disorders, is there's now tools with gene editing like CRISPR to make what we call quote unquote humanized models in the fly. So remove the sister fly gene and replace it with the human gene, and then in parallel test the patient's variant. So it's a very new and attractive tool, and it works. And we've published this for new disease genes, also genes with what we call phenotypic expansions, basically patients that don't quite look like the first cohort of patients that were identified. We've identified drug leads in these cases and used multiple strategies to do this. Um, when it comes to KAT6, which I'll just kind of say, I know we're very interested as a group on the differences between KAT6A and KAT6B, but as I'll explain in the flies, because they're invertebrates, they actually only have one gene that's the sister gene to both KAT6A and KAT6B, because mammals had a lot of chromosomal duplication, and that's why we have two copies or more of genes a lot of the time. And so the fly is a simplified model, but again, as I alluded to, when we look at the amount of variants that we see in the patients for both disorders, or all three disorders, I guess, there are a lot of missense variants. We know they're mostly de novo and heterozygous, and a lot of presumed loss of function or haploinsufficiency. And to my knowledge, and I'll be corrected in the scientific meeting, there is this clinical worsening or severity of these later truncations. Truncations just means either a frame shift or a nonsense um, that seem to be worse, and there's no concrete evidence that I've seen yet for dominant negative, but that's presumed because there's no, for the scientists in the room, nonsense mediated decay. Uh, that peptide will still get made and may have some toxic function. And so the point is, though, we lack functional studies on a lot of these variants. And so the objectives of this small sort of 25K Canadian, which is probably what, 18 American, um, that I received a couple years ago, what can we do with flies with our proposal? One, we wanted to make a mutant fly. Mutant flies were made before to knock out that sister fly gene, and it's called Enoch. So if you hear the word Enoch, that just means KAT6 in flies. And we wanted to use this new mutation that allows us to see where it's expressed. And we wanted to show it's actually expressed in the brain, even though 24 years ago, this gene Enoch in flies was already published, called Enochi mushroom, just because it's important in the mushroom body of the fly brain. Flies have a brain, 200,000 neurons, and it's important for learning and memory. So we already knew it was important in the brain. Secondly, we wanted to assess obviously the developmental role, but also what's the role of Enoch in the fly brain in the adult? And we have a lot of sophisticated genetic tools that we can just remove ENOC in neurons of only the adult flies. Let them develop normally and see what happens when we remove ENOC in the adult brain and attempt rescue with a drug called nilotinib that I'll explain in a few slides. And then lastly, can we assess these variants by overexpression? So this was a partnership between KAT6A, and in Canada, there's money for rare disease for these sort of catalyst grants from the Rare Disease Models and Mechanisms Network. So we successfully made a mutant, which, as predicted, was homozygous lethal, just meaning flies, just like us, have two copies of every gene. If they're missing both copies of ENOC, you don't even get an adult fly. They die as embryos. And these two, ENOC1 and ENOC2, are classical mutations that were made decades ago that are also lethal. And when you put them over each other, everything's lethal. It just means we made a clean mutation, you know, it's working. But what's great about this mutation that we made, it allows us to see gene reporting. And what you're looking at is actually not the brain of the adult fly, but the brain of the larva. So you put these flies on basically molten molasses food. They lay their eggs. Embryos develop into larvae, and they chew through the food and eventually come out of pupa. And so they're just like a butterfly, right? Just not as graceful, I guess. But it lights up in the brain, just showing us that this is a gene reporter that, of course, ENOC is expressed in the brain. We kind of predicted that this would be the case. And single cell sequencing databases show that it's expressed in the adult brain and in a lot of neurons. So this green here is just showing you clusters of cells from the fly brain, and these are all neurons. And these two drumsticks over here are the supporting glia. ENOC is expressed in many of the neurons and some of the glia, but it's mostly enriched in neurons. And so what did we do? So first we knocked down, so we already knew that homozygous loss of ENOC is lethal. You don't get a fly. But also, if you knock down by what's called RNA interference, ENOC just in neurons during development, you also don't get any flies. Now, there's different temperatures here because the higher the temperature, the more we knock down the mRNA of ENOC. So at room temperature here, 22 degrees Celsius, for those who know this system, that is room temperature, so about 72 Fahrenheit. And so you do get a few flies, but those flies die pretty quickly. So in the lab, fruit flies as adults can live for 60 to 80 days. Okay. And so this was true in males and females, with a little bit more females surviving. Uh, these flies that did come out of pupa, they had wing defects. So we're knocking down ENOC in all neurons. This is pan-neuronal. So there are nerves that go into the wing. And so ENOC is somehow required for the wing to develop properly as well. And of course, likely due to the wing defect, these flies at 2 days old as adults cannot climb the side of the vials at all, whether they're male or female. And so this led us to this sophisticated strategy of let's induce the knockdown, meaning let's only remove the fly KAT6, ENOC, from adult neurons. Let them develop normally. And then once they come out, we put them on food with a drug that causes this genetic system that we use to knock down ENOC. And you can see in the purple, when we test their ability to climb, they take about twice as long to climb, whether they're males or females, at 20 days old. So um, this tells us that ENOC in the adult fly brain, even after development, has a role for proper neurophysiological behavior. And so that led us to literature, and actually another Canadian group published a paper in 2018 where they took mouse cortical neurons, put them all in, 200 different wells of a dish, and treated them with different brain penetrable drugs that are either approved or in some phase of clinical trial, and then did what's called RNA-seq that was mentioned before, and looked at how these drugs change the transcriptome, or how these drugs change gene regulation. What was really intriguing to me, if I look up KAT6A, the mouse gene, or KAT6B, the mouse gene, the top drug that increases the levels of mRNA of KAT6A or KAT6B is this drug called nilotinib, which is used for leukemias and is a kinase uh, inhibitor. And it's also, if you look it up on Wikipedia, it's sort of there's something bubbling about Parkinson's trials and other neurodegenerative disorders. However, unfortunately, in our model, when we remove ENOC in neurons during development, and we get a lot of death or just flies that don't survive very long, nilotinib, at least at the first initial concentrations tested with our funding, failed to rescue either what we call the Mendelian ratios, but basically just means how well they survive coming out of pupa. And I had showed you earlier that they die pretty quickly, and our control and our drug does not rescue those lifespan defects. So what next? First, we're going to start with our adult-specific phenotypes in the flies, which was the climbing problem. So I showed you at 20 days, they take about twice as long to climb. The threshold for rescuing something like that is a little lower, and we just want to test that first. And of course, we want to try different concentrations. I've been in touch based on the last virtual research meeting with Monica Denton, who has sent over luteolin, which is another drug they're interested in testing. And of course, throughout today and at the scientific meeting, the models I'm explaining now are definitely conducive to testing other drugs, which we could definitely do. And so the last part of my grant was I looked at when this call was made, I looked at what was sort of published as far as variants, and this was 2 to 3 years ago. And, we didn't have all the money in the world to make everyone's variants. So I chose truncations in purple that were kind of spaced out. Particularly, I was interested in these sort of later truncations as well, in both genes, as well as hotspot variants that were de novos in multiple patients, and tried to pick missense variants that were in different domains for both of these. We successfully made transgenic flies that'll only overexpress now we're overexpressing human KAT6A or KAT6B with the variant or not, uh, in a tissue type way in the fly. And so this slide sort of explains that is when we make these flies and they get, embryos get injected with a plasmid, just a circular piece of DNA that has our human gene. It incorporates in the genome, but will not express yet. I'll tell you when it expresses. But we put in the reference, which just means the human wild type, the quote unquote typical sequence of KAT6A, KAT6B, and we put in the point variants that I just explained yesterday. Not yesterday, in the previous slide. The trick comes in, how do we do tissue-specific overexpression? So this fly is not expressing anything yet until we cross it to a female. So most of the people in my lab are collecting female virgin flies from these lines. That just means young flies that haven't mated with the males they're hanging around with. So we can mate them to males here of this line. And in the progeny, the next generation, they will carry both the promoter for a specific type of tissue as well as this transgene that will allow the overexpression in this tissue. So we can do this ubiquitously. That just means in all cells of the fly, we overexpress KAT6, A or B or variants, developing eye, wing, neurons, glia, etc. There's 8,000 tissue-specific lines available in Bloomington, Indiana for $20 a piece. So that's what helps with fly work. And so this is not KAT6, but this is just an example of what we see when testing variant function. So when it comes to overexpressing a human gene in all cells of the fly, 30% of the time, it kills the fly. You're pumping in a lot of exogenous or topically expressing a human gene, and sometimes it kills the fly. You don't even get the fly come out of pupa. But what's interesting about this assay is if you test a single point variant in that same gene, and all of a sudden you get flies that are alive, it's kind of the opposite way of thinking about things. If normally the function of the human gene when overexpressed in the fly causes toxicity, and you lose that toxicity when testing the patient variant, it's likely some type of loss of function, that variant. But we also see the opposite. We can overexpress a lot of a human gene in all cells of the fly, and the patient's variant you don't produce a viable fly at all. And so it's some type of gain of function. We do not know yet whether it's dominant negative, neomorphic, meaning having some new function, or hypermorphic, doing what it normally does, but at a high rate. But at least we know it's under that umbrella. And this is when we use the eye and the wing of the fly because they're dispensable tissues. You can produce a fly that has no wings or has no eye, and they still come out of pupa. So here's a case where a variant causes problem to the eye development, so it's likely acting as a gain of function. And here is an example where the reference of a particular gene is causing a wing margin loss, and the variant fails to cause that, it's likely loss of function. What's great about these tissues is, as I alluded to before, we know a lot about the signaling pathways involved in wing development, eye development. And so even though these are overexpression assays, it can give us hints on what might be going on from a signaling point of view. So the next few slides are some of the data from our variant testing, and it's still preliminary, and there's a lot of variants to test. So this is when we overexpress KAT6A in all cells of the fly. We had a feeling because this affects transcription, when we overexpress reference in the fly, it'll likely kill the fly because we're expressing it in cells that probably don't necessarily want KAT6 expressed. Gene transcription will be changed during development, and you won't get an adult fly. And this is true in males. And what it allows us is to test the variants. So the first three variants you see are an early, middle, and late truncation. And so what was intriguing and was the differences between them in the sense that, yes, a frame shift at amino acid 16, you're barely producing anything, is now producing viable flies. So what does that mean? It means that likely it's a strong loss of function variant, which makes sense. The reason we do this at different temperatures again is that higher temperature, the more we overexpress. So previously, the higher temperature, we used RNA interference to knock down the fly gene. Now we're doing higher temperatures, the more we overexpress the human gene or cDNA. This truncation in the middle was still somewhat functional, which was a bit surprising. And then this latest truncation, which was acting as a loss of function or producing viable flies at 25 degrees, if we ramp up expression, it's still similarly functional at higher temperatures. So there's more to do, and we need to do lower temperatures to kind of take all data together to kind of figure out what's going on between these truncations. But it is telling us that the truncations are acting differently from each other. That's all we can say right now. And then we have a slew of missense variants. And all of these missense variants seem to be just as functional as the reference, meaning they're still killing the fly. So we cannot really distinguish function yet. So we have to lower the temperature to 22 and 18 degrees Celsius to see if we can tease that apart. The exact same thing was done in female flies, and you see a similar scenario. So no sex differences. When it comes to KAT6B, it's a different story. So KAT6B, when we overexpressed, did cause a lot of lethality, just the reference in flies, but not as much as KAT6A. KAT6A, you barely get any flies at all, whereas KAT6B reference compared to our controls, where you would expect 100% here, is quite less than 50% of expected. So there's some death there. The truncating variants, this time we tested 4, all seem to be acting as loss of function. When we increase temperature, they may still retain some function compared to controls here. So we need to tease that apart. To date, we've tested 4 missense variants, and a few of them here, the first 3 seem to be loss of function, whereas the last one seems to still be killing the flies like reference, potentially even worse. So we don't know yet. It's not statistically significant. We have a few more trials to do. So we'll have to see whether this is different from KAT6B reference or not. But we can say that it's at least functional, potentially gain of function. And a similar scenario was seen with the female flies. However, I will state that that last variant here may actually be a gain of function in this assay. Because why is it gain of function? Is because KAT6B reference is not killing so much in the females, and this missense variant is killing more, meaning it's having some extra activity, right? And it's great to do ubiquitous, but you don't really learn how to really figure out the spectrum of variants completely. So we turn to the wing. And sure enough, when we overexpress KAT6A in the wing, it causes this very stereotypical phenotype. And most wings look like this. They lose these bristles here, and they actually have a blister here. The wing is basically two sheets of cells that need to merge. There's probably some change in adhesion molecules due to the overexpression, and you get this blister phenotype. It's kind of hard to see 2D, but there's a blister there. And so the three truncations that we had, even though they had different functions with the ubiquitous expression, in the wing, they all seemed to be loss of function. But if you look very closely, the wing is very stereotypical in our controls. Even this A16 frameshift has these minor wing vein defects. And so this is very preliminary, and there might be some defects here. So even though we're calling these preliminarily loss of function based on this assay, there might be some still retained function in these truncations. And then in the missense variants here in red, they're all showing a variety of function, some potentially gain, and we need to correlate to our ubiquitous overexpression, and we need to quantify. So we need to quantify, are the wings smaller? Some of them do look smaller for sure. Uh, how many have blisters? How many have bristle loss, etc.? How many have cross vein defects? And so this is what we'll be doing over the next couple of months. Intriguingly, KAT6B did not cause a wing phenotype like KAT6A did. But again, there may be minor changes uh, to the wing that we'll look at, and we'll still cross all the KAT6B variants because we may catch some gain of functions there as well. What Now, my lab stumbled into KAT6A because of this funding call, and I kind of had a hunch that overexpression in flies would cause a phenotype that we can test all these variants. But in my postdoc, I had a similar gene, IRF2BPL, causes a neuroregressive disorder in kids at the age of 5, something similar to Rett syndrome, but more severe. Um, but when I overexpressed that gene, I had a similar wing phenotype, and we dug in deeper in this model here and showed that actually it inhibits Wnt signaling, which helps the wing develop. And so when you lose that gene, you have excess Wnt signaling. And Wnt comes up in half the autism genes and is clearly a very important developmental pathway and probably has roles in the mature nervous system as well. What's intriguing about the KAT6A phenotype is it's very reminiscent of a disruption of Wnt. And sure enough, in cancer studies, KAT6A has already been shown to be a regulator of Wnt, and may also uh, enhance non-canonical Wnt signaling. So there's a connection there to Wnt signaling. And so from my lab's point of view, it's a gene that, for bigger funding calls to the Canadian government, we want to look at KAT6 plus other neurodevelopmental disorder genes related to Wnt. So in summary, the fly gene is required for flying neurons for viability of the organism. So just knocking it out in neurons, you don't get a viable fly. It has a role in the mature nervous system in maintaining proper neuronal function because those flies have climbing defects. And the fly model can be used to assess variant function by overexpression. So the future directions that we're doing this summer is we want to find some other phenotypes with our adult knockdown of ENOC in flies. So not all KAT6 patients get seizures, but having another assay in flies to induce seizures to potentially test drugs, the more assays we have with a phenotype, the more ability we can test drugs to rescue those problems in the flies. We want to test nilotinib and other drugs in that model I just stated, complete our variant assessment in the wing. There's another that I alluded to humanization, but I don't have time to talk about it, but it's genetically complex. But we do want to attempt to humanize these flies, meaning in the background when ENOC is gone, can KAT6A rescue? And this kind of goes to the question we had in the middle for the last talk. So overexpression of KAT6A or KAT6B causes problems when overexpressed in all cells of the fly. That's bad, right? If we're a little worried that if we can use the Enoch mutant we made to express the human KAT6A or KAT6B, but it will be an overexpression under the same spatial and temporal pattern as the fly gene, meaning whatever fly cells normally express Enoch, they'll now overexpress KAT6A or B. That's what humanization is. If that kills the fly, that means that this is a dosage sensitive gene, and at least in the fly, uh, but would, you know, tell other human researchers to be careful from a gene therapy point of view that dosage sensitivity may be important for this gene. Um, and then finally, if we can confirm that indeed Wnt signaling is affected, it is a druggable pathway. Carefully, because it's important. But there may be some drugs that can modulate the Wnt signaling pathway that could be employed in our studies and potentially translate to clinic. And we are, as alluded to before, we're very interested in drugs that are already approved so they can be repurposed. And so those are the kinds of drugs we ideally want to be screening. So I'd just like to thank all the Enoch work was done by a co-op student named Harsimran Kaur. And Zoe Goldstone Chabert is my genetic counseling student that actually is our first genetic counselor doing wet lab fly work. And it's working out. And she definitely is interested in coming here next year if there's a meeting to present all of our final variant assessment data. And Hannah Spring is a summer student helping her. So thank you. I'll field any questions at the time. Thank you.
Everybody, any questions? We'll have one more presentation before we have a 15-minute break. So thank you all for there.
You had said before that the flies in the beginning had a problem climbing up the wall, correct?
When we knock it out in neurons during development, yeah.
Um, or not or
So it was two different paradigms. So one is we're knocking out Enoch in all neurons at all times, so both development and in the adult. And those flies have a wing defect, and immediately at 2 days they cannot climb, um, and they die pretty quickly. The other paradigm was nothing's happening to the flies genetically until they come out of pupa and are adult flies. Then we knock down Enoch in neurons. And at 20 days, they have a climbing defect. What we have not done is checked at day 5. Do they have a climbing defect at day 5? And is this a progressive problem or is it immediate? So we still need to do that.
So they could apply over on the platform.
That's true. And mosquitoes. Yeah.
Are there agents other than kinase changes that have been shown to have impact there?
Yeah. Yeah. So Wnt is obviously it is a complex. There's more than now 3 pathways involved in Wnt, but from a drug point of view, yes, GSK inhibitors to enhance Wnt signaling uh, have worked. Lithium chloride uh, is a bipolar medication that also enhances Wnt. However, my hypothesis is that there's excess Wnt because overexpression of KAT6A is inhibiting Wnt, so the opposite loss of KAT6A probably leads to excess Wnt. So from an inhibiting point of view, a lot of them are CK1 alpha agonists. So the preclinical stuff are these XAV939 in the cancer field, and there was an old drug called Pervinium. It doesn't get into the brain, but what we've been looking at is drugs that are not bona fide Wnt inhibitors, but have been shown to inhibit Wnt in some way. So drugs like Sulindac, even curcumin can inhibit Wnt. The only other connection, and we'll bring it up in the scientific meeting, in my other gene, we also see mitochondrial defects and fragmentation, and there's a connection between Wnt and mitochondrial biogenesis. And we've been testing in those models that we'll probably bring to the KAT6 models, these raw, super potent antioxidants like N-acetylcysteine amide. The amide slapped at the end so it crosses the blood-brain barrier, CoQ10, coenzyme Q10. Um, but I do have a list of Wnt modifying drugs if you're interested. Okay.
Okay. So, okay, now let's get from Amy Serrano lab at the university.